Next part in the viruses is ICTV classification of viruses. This is a classification system for the viruses, whether it is animal virus, plant virus, or any microbial virus. So all these viruses, they have been classified into numerous families based on their genome structure, that is whether it is RNA or DNA, and in RNA and DNA whether it is double stranded or single stranded, whether it is linear or coiled, its life cycle, then its structure, morphology, structure of capsid, the complex structure or simple structure, envelope viruses likewise, and the genetic similarities. So all these characteristics are very useful for the classification of viruses. So these families have been designated by International Committee for the Taxonomy of Viruses, which is called as ICTV. ICTV, it is a committee that decides the classification criteria for these different viruses. So this is an agency, and this agency is responsible for standardization of the classification of all the viruses. So it was in 1971 the ICTV developed a uniformly classification system for the viruses, and since then the number of viruses and their taxonomic categories have continued to expand. So this is just like classification of bacteria. There are millions or billions of bacteria whichever the bacteria are discovered are classified according to Burgess Manual of Determinative Bacteriology and Systematic Bacteriology. So in case of viruses also, there are millions of viruses which are classified using this ICTV classification system. So whatever the viruses which are discovered, they are only classified. There may be undiscovered viruses also. So it was the ninth report which was prepared in 2012 of ICTV. Almost only 2,284 virus species have been described and placed in 6 orders, 87 families, and 19 subfamilies, and 349 genera. So in order to place the virus in a taxonomic group, taxa means it is group, taxonomic group, the ICTV considers a range of characteristics. So what are these range? The first characteristic is what is the host range of this virus, means whether this ability of this virus to infect a type of host cell, whether it is prokaryotic cell or eukaryotic cell, whether it is animal cell, plant cell, or it is bacterial cell, or algal cell, or fungal cell, or protozoal cell, etc. Second, morphological features of the virion, that means the structure of virion. So we have seen earlier the structural characteristics of virion, the shape of the capsid, envelope of viruses, presence or absence of envelope, shape of capsid, or the shape of nucleocapsid, etc. Then the another characteristic to be considered for classification is nature of genome or nucleic acid present, DNA or RNA, as we have seen, single stranded or double stranded, positive or negative strands. Then within these parameters, additional features are considered. For example, the length of tail of a phage, or the presence or absence of specific genes in genomes of similar viruses. So this is basically we make use of different features or different characteristics, distinctive characteristics of viruses to group them or to classify them. In ICTV system, the names of virus, order, families, subfamilies, genera, and species should be written in italics with the first letter capitalized. Virus, order names end with viroids. Virus family names end with viridae. Subfamily names end with virini, and genus names end with virus. So all these are the parts of nomenclature system. Nomenclature means naming system. So ICTV it established rules for virus taxonomy, such as no person's name should be used in nomenclature. That means in name, the name of the virus is not given uh, on the basis of the name of the discoverer, which happens in case of bacteria but not in case of viruses. So that is the point to be considered as. However, it may include the name of the disease it causes. An example of this nomenclature system is shown in this table. You can see in the first column taxonomic group, which shows order, family, subfamily, genus and species. Then second column shows the suffix. For order it is virals, for family it is viridae, for subfamily virini, genus is virus. And there are examples. For example, the name of the order is Mononigarales, Gavirales is the name of order. Then family is Paramyxoviridae and subfamily Paramyxivirini. The genus Morbillivirus and the name of the species measles virus. This is an example for the classification of measles virus. That is all about the viruses. The next type of organism is viroids. Now, eukaryotic cells are complex, then simpler ones are prokaryotic cells, and more simple than prokaryotic cell, bacterial cell, are viruses. Viruses are basically made up with proteins with nucleic acid. Viroids are much simpler than viruses. Viroids are much simpler than viruses. What are viroids? Viroids only contain nucleic acid. So they are much simpler as compared to viruses. It was discovered in 1971 by Danier, who discovered an infectious RNA particle smaller than viruses that causes diseases in plants, and it was called as viroids. It is very interesting to know that it is only single stranded DNA which is responsible for causing infection or diseases in plants. So a viroid, it is a circular molecule of single stranded RNA without capsid. That is the difference between viroid and virus. And it has double stranded regions which are shown in the figure and consist of only 250 to 370 nucleotides. So it is a short chain of nucleotide RNA, similar to virus in that it can reproduce only inside a host cell as particle of RNA. That is a similarity. So it also needs only a host cell in order to multiply. The size of viroid is only about 1/10 the size of smallest plant virus. This is the structure. Here you can see it is single stranded RNA molecule which is circular, and in few regions you can see it is folded, and you can see the double stranded regions due to the formation of hydrogen bonds. Second one, viroids cause several economic important diseases of plants. So single stranded RNA are responsible for causing diseases in plants. For example, potato spindle tuber viroid, it is responsible for causing the deformations in the edible part of potato, that is tuber, causing it to become long and pointed. So it also causes stunting of the growth in case of tomato plants. Cucumber pale fruit disease and exocotis disease of citrus tree are also caused by viroids. As few of 10 molecules of single-stranded RNA are enough to infect a potato plant. That is all about the viroids. Other simple type of organisms are prions. So in contrast to viroids, prions are only made up with proteins. So these are infectious, low molecular weight protein particles which are responsible for infection. These are responsible for infection in animals. So they thought to cause about a dozen of fatal degenerating disorders of central nervous system of humans and other animals. So it is very important. It has medical importance that it is responsible for the diseases of central nervous system, and these are caused by only protein particles. Unique, it do not contain nucleic acid. A very important difference between prion and viral. It does not replicate itself, so it has to enter into a host cell. It changes now exactly how it replicates. So whenever it enters into a cell, it combines with another host protein, and that host protein, after combination, it becomes a prion, which is called as PRP, that is prion protein, and this prion protein is infectious. When a prion enters host, it forms more prions from the cell's proteins. So that means it combines with several other proteins and forms prions. These prions accumulate, aggregate into fibrils and cause diseases. A neurologist in 1982, Stanley Prociner, he proposed that proteins can cause neurological disease called as SCAPI. So he called it as prion. He was awarded a Nobel Prize in 1998 for his vital research on prions. These are the examples of certain neurological diseases which are caused by prions. First one is scrappy. So this is a disease of animals, so domestic animals such as sheep and goat. Due to this disease, these animals they lose coordination of their movements and scrap or rub their skin and eventually cannot walk. Second disease caused by prions in humans is kuru. It has been only found in the four. It is an eastern guinea pig tribe. This tribe has a custom of consuming dead animals' brain. They and their children were infected by handling the deceased brain tissue. Now this tradition has been eliminated and therefore incidence of kuru has been decreased. So these people come in contact with diseased brain tissue which contains these prions, and these prions when they are consumed, these consumed prions then combine with the proteins in the human body and it multiply and further leads to this neurological disorder. So it was in 1976 Carlton Gajudusk won a Nobel Prize for his work on Kuru disease. Another one is cow disease called as mad cow disease. So it may be spreaded to humans due to consumption of diseased animals. And the fourth one is Creutzfeldt-Jakob's disease called as CJD. It is also a numerological disease. So it is first discovered by Hans Gerhard Creutzfeldt and Alphonse Maria Jacob. That is why the name is given on the name of discoverer.